This is Will to Win, Overcoming Long COVID. My name is Jillian Angeline, and my goal is to raise awareness about the hundreds of millions of people worldwide who are struggling in many cases for years after contracting COVID. I know about that myself. 5.5 years after getting long COVID back in February of 2020, I've undergone 6 surgeries, and I'm on a road to recovery and healing, but it hasn't been easy. Today's guest is Professor Kell out of the University of Liverpool. He's a systems biologist and a biochemist. Of course, these episodes are for informational and educational purposes. None of this constitutes medical advice. Please connect with your medical professionals before trying anything we discuss here on this episode. Let's get started. What is long COVID in your mind? I could give my own definition, but you probably have a better one.
So long COVID, and indeed any other post-viral disease, is something that is associated with having been exposed to, in this case, SARS-CoV-2. And some people just get better afterwards, but other people, and the definition is usually used to be 3 months after your infection, continue to have symptoms that go from unpleasant to debilitating, and it varies very strongly between individuals. And that is what we normally call long COVID. The commonest symptoms are fatigue, what is sometimes called brain fog, and what is called variously post-exertional malaise or post-exertional system exacerbation, where not immediately, so fatigue is you get tired just as you're doing some exercise, even trying to go up the stairs, whereas PEMPESE is something that happens a day or two afterwards. And we have a couple of mechanisms now that we think can explain what's going on there. It's different from fatigue, of course.
And you've been part of a study with many other doctors that focus on the fact that there may be some clotting as well in the musculoskeletal tissue. And I thought that was fascinating because for me, I've tried workout classes. I've tried to do whatever I used to do. I've run half marathons. I've run 5Ks, 10Ks, you name it, pre-COVID. I can't work out the way I used to yet. I'm hoping after all these surgeries, like the one I recently had, as you can see from this bandage, that I'll be able to get back to that. But tell me a little bit about what you all figured out when you did research on more than 40 patients.
So we have to go back a little bit to our discovery of anomalous blood clotting, which goes back as part of a long-standing collaboration I've had with Professor Risha Pretorius from Stellenbosch University, who was looking, when we first met on the internet originally, at the role of the metal iron in affecting the ability of blood to clot. And she looked at an electron microscope. Normal blood clots look like a plate of spaghetti, as in spag bol. These clots look like as though you'd partly boiled the spaghetti and let it clag together into a rather disgusting mess, which was referred to at the time as dense matted deposits. And we both had an interest in iron metabolism and started working together. And a few years later, we discovered that these anomalous clots were actually what is called amyloid in nature. That is to say, the proteins involved, which is principally fibrin, going to fibrinogen, adopt a structure that is completely different, which makes them resistant to being got rid of. And you can stain that with a stain with what's called amyloid, thioflavin T, and they fluoresce like bilia. So we discovered that this is what was going on, and that can then, when we fast forward to discovering that these things are happening also in both acute and long COVID, if you've got things going around your bloodstream, and especially the small vessels, the capillaries, that basically stop the red cells taking oxygen to the tissues, that can pretty well explain all of the very diverse symptoms of long COVID and indeed ME/CFS as well. And we've shown that there's a strong relationship between the presence of these kinds of diseases and the anomalous microclots. And we've also shown that the spike protein alone is sufficient to trigger the transfer of a clot type from a normal clot into the anomalous, we call it fibrinoid microclots. And so these things we think can explain a whole load of things, including the post-exertional malaise, which we'll come to, and other things like autoimmune responses and postural POTS, it's called. I just won't use the long words, which cause people hearts to beat much faster when they try and do some exercise or bend over.
Or just standing up.
For us, the microclots give a very straightforward explanation, because what's happening is that you're getting the blockage, and what's the heart trying to do? Well, it's going to beat faster because it thinks that there's not enough oxygen going through, so you don't have to invoke anything other than normal physiological response to describe POTS. So in the recent study, what was done, it was led by Mark Fye from University of Derby, was to have people do some exercise under carefully controlled conditions, because no one wants to induce PESE. And then he looked at the size distribution of the microclots. And what he discovered was they essentially fragmented when you did exercise, which of course creates a lot more of them. And if you think a clot of diameter 10 has a volume 1,000 times more. So if you have a diameter 10 and then you make them all down to a diameter 1, you essentially get 1,000 times more of these things. And so it was considered that that would also be a significant contributor to post-exertional malaise. Although the hypoxia alone, when you lack of oxygen from doing these exercises, when you put the oxygen back, which is called reperfusion in the trade, you essentially cause an injury to cells because of this so-called hypoxia reperfusion injury, and that is what causes also the post-exertional malaise with a delay of a day or two, because that is the time it takes the cells to die following the insult of the hypoxia reperfusion injury. So that's the broad story, and the observations are very clear-cut. The difference was very large.
Well, I will say that when I did see that was posted, I think on LinkedIn, when I first noticed that one of the doctors shared that study and your name was tied with it, when I clicked on the study, it's not peer reviewed yet. Looks like it will be at some point soon, I hope. I went straight to the conclusion, because for me, I'm trying to look at the positive. So I click on the conclusion and I go, what does this mean for the future? What does this mean for all the long haulers who want to be able to work out again? And what I loved is the one line that said, this kind of research will inform future rehabilitation. And that kind of phrase, or therapeutic possibilities. And that gives me so much hope to be able to see something like that, because then I know that if there are people out there like you that are doing this research, it means that you can inform doctors, physical therapists, you can inform us patients. So let us know, hey, this is what we're seeing.
The scientific consequences are very clear. Get rid of the microclots. And in all kinds of diseases, we found that normal healthy controls have next to none of them. And people with various kinds of diseases, especially long COVID, have loads of them. And Caroline Dalton, who's at Sheffield Hallam, did a similar study in which she looked at the relationship between the number of the anomalous microclots and the severity of the disease. And she also found a good correlation with that. So we're not the only people finding this by any means. And of course, the other control we did, when I said that we could prove that spike protein alone, when added to normal plasma, caused the anomalous clotting, we also did it for the later Omicron variant, and that was much less good at doing that, and it is also much less virulent. So that's an important control that tells you that the microclots are on the disease pathway, because if they'd been the same, whereas the thing is less virulent, you wouldn't draw that conclusion.
And I got one of the very strong strains that was the first to come from China. And that's why I think that I wasn't as bad of a shape as I was, needing, 6 different surgeries to try and fix those issues in my veins.
And we shouldn't ignore viral persistence, because the question is the virus lurking in people's body when they have long COVID? And the answer certainly is sometimes. Eventually, if you get rid of it, of course, all will be well. But we're pretty sure that in many cases, the virus persists, hidden away in various places. And of course, it can then keep creating more spike protein and more anomalous clotting. So the trick is to get the anomalous clotting. That's the trick. If you can do that, you should solve it.
Because COVID is a clotting disorder. And a lot of people early on were talking about being a respiratory issue. It is a clotting, but it has to do with the vascular system.
100%.
And I think once that needs to get out there, a lot of people don't realize that. And the regular public, I think, when I talk with people about long COVID, a lot of people don't realize that people are still dealing with it. We have 400 million people in the world feeling long
Oh yeah,
COVID
yeah,
right
yeah,
now?
yeah. My personal view is that actually, because for practical purposes, everyone has been exposed, everyone has some element of long COVID, but of course, some people have symptoms that are so minor that they wouldn't even notice, whereas others, as mentioned, have very debilitating symptoms.
Tell me about the importance of collaboration between researchers, scientists, and doctors from across the entire world. How does that feel for you to be working with people, with Dr. Riccia Pretorius out of South Africa and so many others from one country to another country? This is really a global teamwork effort.
So typically, scientists work with people they get on with and who share similar views to them. And as you mentioned, I'm a systems biologist, and people who think like a systems biologist are the people I tend to work with and collaborate with over the years. If you look at the sort of people I've published a lot with in different fields, when I worked in yeast, I worked with someone called Steve Oliver, and when I work in other things, I have a lab in Denmark. I work with someone called Irina Borodina, who I've known for years,
Mhm.
and these people think like systems biologists. And a new collaborator here is Bernadette Moore, who is actually a professor of nutrition, but her first degree is electronic engineering.
Oh,
And,
wow.
and you know a fellow called Jordan Vaughan, who's in Birmingham, Alabama, who's been doing a lot of great work in helping people. And I asked him, how come, Jordan, you get our stuff? Because he understands our stuff and is implementing treatments designed to get rid of
Mhm.
microclots and so forth. And he said, well, my first degree is in engineering. So he doesn't think like a typical medic. He thinks like a systems person. You can't design a car if all you focus on is the wheels, for instance. So you have to think like a systems biologist. And I could say people like Lee Hood have been promoting this idea at the Institute for Systems Biology for many decades. But that is the right way to think about it. And of course, if you look at things like traditional Chinese medicine,
Mhm.
in those days, doctors were paid when you were well. This contrasts significantly with the circumstances now in which doctors get paid when you're ill, which, if you think, is not actually the logical point of view if their job is to keep you well. And so the traditional Chinese medicine people recognize what we would now call personalized medicine. Everyone is an individual, and you have to treat the individual. And you don't ask which disease does the individual have, you ask which individual the disease has. And that's quite a different way of thinking. And coming on to Chinese medicine, there's a concept core to it called blood stasis. The term was used also by Virchow in the 19th century in the West. And when you look at what the Chinese have in mind when they talk about this, as the name would suggest, it means the blood isn't moving around as effectively as it should, which of
Mhm.
course is smack on exactly what the microclots are doing to stop it here.
Absolutely.
And we published a review on this, which I'm sure you can cite a
Mhm.
link to at the end. And the most common treatment for blood stasis is a cocktail of 11 herbs called shui fu zhu yu. Shui means blood in Chinese. And we're pretty confident that a number of the things in this are actually preventing the anomalous clotting,
Mhm.
but not preventing the normal clotting. And we're working on that now. But if you look into these areas
Mhm.
and measuring the microcirculation, so in TCM, Chinese medicine, tongue diagnosis, where they look at the tongue, they're actually looking at the microcirculation. And you can also look at
Oh, wow.
in the hands just below the nails at the so-called nailfold capillaries. And they will tell you a lot about how the microcirculation is doing. And in a more extreme example, if you look at sepsis, which is commonly, but not exclusively, again, an infection, which leads to a hyperactive immune response, which is what actually causes the septic shock and the sepsis. One of our colleagues in the University of Liverpool looked at our methods and wondered whether or not the microclots were related to whether you came out of the intensive care unit from sepsis dead or alive. And he had a freezer full of blood samples, and of course he knew whether people had survived or not. And he asked the question, if you're above a certain fractional number of microclots, do you tend to come out dead or alive? Answer, yes, you can predict it with an odds ratio of 5-1, which in medicine is enormous. You get a paper in a fancy journal for having something that's 10% better. This is 500%, 500% better. So it was very clear. And also there's a particularly nasty version of this called disseminated intravascular coagulation. And the odds ratio for that, the microclots were predicting was 51. So it was absolutely clear that the microclots are involved in this sort of thing too. And most recently, we've actually been looking at what happens after someone has had an ischemic stroke.
Mhm.
And what happens nowadays, so an ischemic stroke is when a blood clot blocks up a major part of your sort of, central nervous system, commonly a main artery. And the assumption had always been that some clot sloughed off somewhere from elsewhere in the system, went round the body
Mhm.
through small arteries and somehow magically blocked up the biggest artery of all, which if you think about it, doesn't make a whole lot of sense. So
Mhm.
what we recognized was probably happening is that our microclots were accreting to form the big clot. And so what happens after an ischemic stroke is nowadays the surgeon will go up with a little pipe through your artery, up the leg, all the way up to here. And inside the pipe is a little thing like a lacrosse net.
Mhm.
And you use that to pull out the clot that's blocking things up.
Wow.
And then all the way it comes out, you take the pipe out.
Mhm.
Normally, people would just throw these clots in the bin. But
Mhm.
our collaborator, Alakendu,
Oh my
uh,
gosh.
decided that
Don't throw them out.
that's right. He made a clot bank. So we got hold of the clots from his clot bank and stained them for amyloid, and lo and behold, they're absolutely dripping with amyloid, which of course explains why they haven't been got rid of by the normal mechanisms, including people also try and add a recombinant enzyme that's supposed to help get rid of them, called
Mhm.
recombinant tissue plasminogen activator. And this was done in 6 of the 8 people we looked at, and it didn't have any effect at all. So clearly, these things are resistant to the normal fibrinolysis, which obviously is true because they're still there. But when you understand that they're amyloid in nature, it makes it obvious as to why.
Can you do me a favor and just blanket definition of amyloid?
So there are classical amyloidoses, which are proteins that come out of solution that are normally soluble but become insoluble, and they form fibers like the spaghetti. And what we and so the classics of these are Alzheimer's with a protein called A beta,
Mhm.
and Parkinson's disease with a protein called synuclein. And there are a number of what are called classical amyloidoses, none of which have anything to do, were not considered to have anything to do with blood clotting. So what we discovered was that blood can essentially do the same thing. The proteins don't change their sequence, but they change their shape, and then they stack up because this shape, it's a so-called cross beta, this shape just allows them all to stack up to make fibers which are insoluble.
So it's basically a giant chain link fence of sorts
Yeah,
inside the
yeah,
vessels
yeah.
or the veins that's inside the veins, correct?
Wherever it goes,
Wherever it goes
and
in the
including
body.
muscles, but stuff comes out of solution, and it basically starts blocking things up and causing the blood not to flow in the way you would like.
But what you did tell me off camera is that there is some hope with your blood stasis study that may show that people, may be able to find a way to get their blood flowing properly again. And even though, we're not recommending anything for anyone, it's important to check with doctors and all of that. That is a level of hope that I think a lot of people could have in their life to know that there's a way to get their blood flowing again. What does it mean for you to be able to be helping to move forward one step at a time and being a part of something that may give people their lives back?
It's relatively unusual for a laboratory scientist who doesn't normally meet clinicians. I mean, I've done clinical work with my collaborators in the past. We worked for years on preeclampsia, for instance, rather successfully. But yes, it's nice. But that's you're trying to. This is the spin-off. If we don't discover anything, then we're not going to help anyone. So our job is to discover this stuff. And when you've discovered it, you can make use of it. So that's the order of events. And that's why, little plug here, you fund fundamental science, because it's going to give you spin-offs that can then be applied in a straightforward way. So once you know about the existence of the microclots, you can start doing something about them. If you don't know about the existence, you can't do anything. I could give an analogy with Helicobacter and ulcers. So ulcers were supposed to be caused by too much stress and acid, but Marshall and Warren discovered that actually they were caused by a bacterium that we all have, but in this case, it got overexcited and started causing the ulcers, and it was called is called Helicobacter pylori. And so this was totally unexpected. Nobody thought that bugs had anything to do with it. They didn't think that the bugs could survive in the acid of the stomach, but anyway, they can and do, and everybody now accepts that this, and the treatment is antibiotics. And bizarrely, one of the antibiotics is bismuth. And for years, people in the US took a thing called Pepto-Bismol, and the biz in Pepto-Bismol is bismuth. So now you understand why it worked.
I've shared my experiences through the years with doctors like Dr. Brooke Spencer in Colorado and Dr. Jordan Vaughn, with other patients. And, some of them have been bedbound with this debilitating long COVID for 5 years. And they're young. They're in their 20s.
Oh, wow. Well,
So
and people have had ME/CFS for longer than that.
So I can't imagine. I mean, it's just such a joy to talk with you and to know that you're on the front lines doing something about this. To be able to have the ability to even just put out this information to help people get their lives back, to stand back up again, to get moving, to get back to their lives. A lot of people think the pandemic is over. For a lot of long haulers, it's not.
No, and there are still people getting long COVID because the virus is in circulation, and some people are susceptible. And of course, some people become more susceptible having been exposed previously. So it's not a once-off thing.
Well, this of course is called The Will to Win. This is the name of these episodes. What is your will to win this mystery of long COVID? What is driving you to keep going?
Oh, well, of course, what drives the scientist is to find the correct scientific truth that explains whatever the phenomena of interest are, which in the case of a biologist are natural phenomena, and veering in the case of human biology into medicine. But we also do biotechnology. But the aim is to work out what's going on, and then you can work out a therapy to deal with it. And that's the history of medicine in a sense.
Well, my mom always said that for the past 5.5 years or so, I have not been saying why me. I've been saying, how do we fix me? And that's something that, I think my parents are both engineers. So I think that may have been part of that mentality is, there is an efficiency and a goal to solve a problem, whether it be a math problem or a health problem. And this has been my goal, whether I'm laying in bed, in those early days, trying to find those doctors like Dr. Vaughn, figure out what they're doing and figure out how I can meet with them to get fixed. So I think that's so important. What is your next step? You're putting out research constantly. How can people find out what your latest work is?
Oh, well, they can go to my website, which is dbkgroup.org, Delta Bravo Kilo Group.org, and all of the papers are there. And there's also a web page devoted to long COVID, which is dbkgroup.org/long COVID, all lowercase. There's also a Twitter or X feed called hashtag Team Clots, where people who are interested in this sort of stuff tend to put that hashtag in, so you can see that. And of course, people can send me an email if they wish to. Where we're going next, so the microclots we consider to be a structural manifestation of things that have gone wrong in the microcirculation, or they're the cause of it. But they're not of itself a functional measurement. So now we've got interested in functional measurements that we hope we'll be able to correlate with the microclots, but at least to begin with, we've been looking into methods, one of which is called Nailfold Capillaroscopy, that tell you about how good or bad your microcirculation may be, and you can buy a little microscope on the internet that you can put on your cell phone for actually about £25. So not as good as the professional ones, but at least you can get a look, and people are starting to do that. Another two methods which look at the microcirculation, which are based on shining a laser on the target tissue and using some fancy maths, seeing the variation in time and space of the light that comes back. And if something is moving, such as blood cells, you can detect that. And therefore, you can detect how fast they're moving. And you can turn that into an image. And these methods are called laser speckle imaging and laser Doppler imaging. And I've just written an 800-page review with Risha and a Chinese collaborator, setting this out for people as to why we think these could be good methods. And they live in their own little world. They've not really been applied in all kinds of diseases that we think they should be. Laser speckle is particularly, and the Nailfold is particularly being looked at in rheumatology, something called systemic sclerosis, another thing called Raynaud's phenomenon, but by and large not applied elsewhere. And these methods are non-invasive and not quite so cheap as the Nailfold Capillaroscopy, but if you're going to have hundreds of patients through, then of course it pays for itself in a very short time, especially if it tells people what they want to know as to how well they are. And and in this case, it's literally real time, so you can look at the microcirculation in real time.
So there's a protocol that you would put out, and then there are doctors all around the world that have this kind of imaging already?
Mostly they don't have this kind of imaging, but
Okay.
the proposal is that they could and should.
And is this expensive imaging compared,
Uh,
that you can buy on a scientific, website that you're encouraging doctors to go out and buy?
Well, I mean, the people make this instrumentation, and I won't do any free advertising for them. But if you type those words into a search engine, you'll easily enough find the people who make this kind of instrumentation. And it's relatively early doors, which is why I can write this big review saying, hey, nobody's been doing this properly. But obviously, the idea is like a lot of our things, we look prospectively, and we don't want to die wondering.
Mm-hmm. Absolutely. And then that Nailfold Capillaroscopy, fascinating that people out there can just buy a microscope and hook it up to their iPhone.
Yeah, just like that.
And then what do you do with that kind of imaging if you see things under that microscope that's different than what it should
if your
be?
nail folds look normal, and probably it's not too easy to describe what normal is on the thing, but they look nice, and there are websites, and I put pictures in our review about what normal looks like. Then that's good news. But of course, there are all kinds of things that can go wrong. They disappear, so. And that is the cause of a lot of the trouble, that the microcirculation stops working. And so if they've disappeared, or if they're blocked up, or if they don't go in nice curves as they're supposed to, or you find there's little blotches because they've actually hemorrhaged, there's about half a dozen things that can go wrong, and you can detect these very easily. And then, of course, it does require some expertise to interpret in detail, but that's again all for the future. What we're looking for is non-invasive methods that can determine the microcirculation easily and that scale to large populations. That's why we like the Chinese herbs, because they scale. If 100,000 people buy Chinese herbs, it's not going to put a dent in the supply of Chinese herbs.
And it's, relatively safe.
And it's relatively inexpensive.
And safe,
And
I
you haven't
assume?
got to wait. Well,
Okay.
it's been going for 3,000 years, this Shuefu Zhuyu, so the assumption is, and at the moment you don't have to go through the FDA or whatever to use these, because they've been around. I mean, if you had to go through the FDA to eat pepper, as in black pepper, would never pass. But it's been around since long enough that it's considered it's fine for people to eat pepper.
Thank you so much, Dr. Kell. I really appreciate your time. As a long hauler myself, I really appreciate knowing that there are people like you out there collaborating with researchers from around the world to give us all hope that there is a way to fix us from the outside in, and from inside to the outside.
We are optimistic, and it's been a pleasure talking to you, and my fingers are crossed that something that people discover, whether it's us or not, soon helps this dreadful disease.
If you have any comments or suggestions, I'd love to hear them. Find me on Instagram, Angeline_travels, or on X @JLAngeline. I'll see you later.